We're here at the IAS meeting in Vancouver, BC. We're here with John Mascola, who's the director of the Dale and Betty Bumpers Vaccine Research Institute at the NIH. And we really appreciate you taking the time.
Happy to do so.
We just were talking that I've interviewed Gary Nabel a number of years, over the number of years, on the subjects of vaccines, and here you are, the next person in line to fill that noble slot. We appreciate you being here, but you brought to the conference, you were at the press conference, and you talked about broadly neutralizing antibodies.
Right.
And I thought it was very interesting, and I know that I would have loved to have asked a lot of questions, but I don't think they appreciate when you ask, half a dozen to a dozen questions. So this is my time to do that.
Great,
great. So we'll let you start with just a brief overview of what that brings to the table and what you see in the research you're doing.
Sure. As I mentioned at the press conference, the appreciation that people make these types of antibodies during the course of natural infection is something that we've only learned over the last 5 or 6 years, and then the ability to pull the antibodies out of B cells from people who are infected and use them as a clinical product. Mm-hmm. So antibodies bring a mechanism of action, attacking the virus, that's different than drugs, and for 2 reasons. One, they prevent the virus from getting into a cell, whereas most of the drugs work by attacking the virus once it's in the cell. And then antibodies can actually have the potential to kill a cell that's infected, which is also different. Mm-hmm. So there's
it's the first line of defense then.
Exactly, yeah. Yes. So there's a lot of excitement about having really another mechanism, another part of the way to attack the virus.
Mm-hmm. So how would you look at this? This is a virologic, this is an immunologic approach?
I would say it's an immunologic approach. It's taking advantage of the body's natural immune system. The body naturally makes antibodies. People who are infected over a long period of time, the immune system finds a way. It's actually a very interesting phenomenon. Over many years, the immune system finds a way to find the vulnerable spots on a virus. It's not an easy thing to do, and it takes some time, some evolution of the immune system. But if you can then take that antibody out of somebody and use it in a different person, you're taking advantage of sort of the best aspects of the immune response.
So this would be the innate immune response, right? Or
technically, we call this the adaptive,
okay,
right, because it's adapting to a viral infection.
Right. Okay. Right. So this is something that I mean, this is the antibody response is what we classify as the way we identify the virus in the first place.
Correct.
So, what is your next steps to evaluate this process?
Sure. The current step is that the antibody is being given to HIV-infected individuals who have some virus in their plasma, and what we can see is that the plasma variant goes down. The antibody has quite a substantial impact on the virus. So that's proof of concept that the antibody as a drug has an effect on the virus. So the next step is really to ask 1 or 2 key questions. Probably the most fundamental question for the cure agenda, the treatment agenda, is can antibodies do something that drugs can't do by themselves? So we know that even if someone is treated for many, many years, fully suppressed, the virus still remains in infected cells, and what we call the reservoir virus doesn't go away. Mm-hmm. So the key question is, can antibodies attack the viral reservoir, kill cells that are infected, and therefore, eventually, potentially get rid of the virus?
So how are you going to accomplish this? Is this how many subjects are you working with, by the way?
So the phase 1 style studies are done in tens of subjects, mainly to get experimentally proof of concept. Mm-hmm. The larger studies to address these key questions are done in some of the networks that we call them, the AIDS Clinical Trials Group, for example, that can do bigger studies.
But so the first sections are done at the NIH?
The first studies are done at the NIH. Mm-hmm. And once we get proof of concept, then we'll do more controlled studies where we look at a larger number of people and start to really measure the viral reservoir in more detail. Mm-hmm. I think the current thinking is that a single antibody administered over a short period of time probably won't have a major impact on the viral reservoir. But one antibody, or potentially more than one antibody,
a different type of antibody.
A different type of antibody that can attack the virus in different ways, given over a prolonged period of time. And we may have to add to that what people refer to as sort of stimulating agents, something that causes cells to activate and express the virus, and as those cells express the virus, they get killed. So the next step is to do those kind of studies.
How much antibody do you need does it replicate, or it's just,
So
it's infinite?
it's a protein, and we can make the protein in the laboratory by standard methods, and it's pretty easy to make. You actually need, grams of protein. I guess that's a hard thing to quantify, but it's a lot. But it's injectable. For example, it can be given as a subcutaneous injection, and then it circulates in the plasma, usually for days or weeks at a time.
So how would this actually be? Is it fair for me to ask how you would implement this as a treatment? Or, I mean, can you visualize any of that at this point, or is it too
I think
early?
we're beginning to. First of all, I think it's worth mentioning that it can be potentially used as a preventive agent to prevent infection, and we're pursuing that.
I mean, like as a PEP?
Exactly.
Okay.
So, and that's maybe one of its most important utilities of antibodies, which are that they could be given to people who are at high risk and potentially prevent infection. So that's obviously separate from some of the treatment discussions we have here, but that's also important. For treatment, I think the researchers would take the best tools they have available. So, to use some of the more technical language, a latency reversing drug, which kind of makes the virus wake up and be stimulated, together with the best antiretroviral drugs, together with antibody, and see if that combination can then lead towards a functional cure.
So, so you don't know how you would stage this or dose it or any of that right now, but that's kind of not really important right now.
Well, those questions are exactly what are being addressed in the phase 1 studies.
Oh, really?
So how much antibody should we give? How much is, if you give it, how much is in the plasma? How long does it last? And therefore, how would you design a study like the one I just talked about, where you're really trying to have an effect on somebody?
What I'm thinking is, like, would it be a boost, an initial introduction, and then a booster, or whatever, something like that? I mean, I don't. That's guess where I'm going. But
right. So as far as if you're asking, for example, how would you give it dosage-wise? An initial high dose, you really want to get a lot of antibody in the system, and we don't exactly know, but it could be administered as infrequently as every 2 weeks, or every month, or every couple of months.
For a given period, or
we probably over a period of months or a year to really,
to make sure it's done.
Absolutely.
Are you working collaboratively with anyone right now, or are you going to wait on that? Because there's so many collaboratories right now that you could.
We have
probably tune in
several,
with,
actually, many collaborations mainly with groups that have clinical trials expertise. So the Military HIV Research Program, there's a group at University of Pennsylvania that has done many treatment trials, the AIDS Clinical Trials Group. Those are 3 of the main ones.
Right. Are you working with any academia that at the current time, or are you just doing everything in-house for the?
One of the nice aspects about being at the NIH is that we can do the initial studies in-house at the NIH to get sort of the critical data needed to design the bigger studies.
Well, I appreciate your offering this up to us, because it sounds exciting, and it's yet one more piece.
Exactly.
Of the compendium of whatever we'll end up with at some point, at least experimentally, until we learn what's going to work best, I guess.
Exactly.
But I appreciate your time.
Thank you.
Thank you very much, John.